the opportunity to share my research results for you, and we already had a very good introduction from Huang on Parkinson's disease. So I also stick a little bit to Chinese medicine, and I talk about this during my talk. So the title of my talk is Telomerase Activators Improve Motor Function and Alpha-synuclein Pathology in a Mouse Model of Parkinson's Disease. So just to let you know, I was a lecturer in aging research at Newcastle University, but I'm retired now and I'm still a visiting lecturer there. So I was at this university for 20 years. All right, so for those of you who are not so familiar with telomerase, so telomerase is best known for its canonical function, which is in telomere maintenance. So it's important that for the catalytic function, you need the telomerase reverse transcriptase, which is shown here in green. And so I will talk in the remaining of my talk mainly about this protein, because it has also non-canonical function where it does not need the RNA component, which is important for the telomere synthesis. Yeah, so it's been known now for 10, 20 years that there are non-canonical functions. And one is that it can leave the nucleus where it normally resides and works on telomeres. So you can see immunofluorescence staining with an anti-TERT antibody here in a cell type where we overexpressed the TERT subunit. It's normal fibroblasts, which normally don't have telomerase. If you now stress those cells either acutely with hydrogen peroxide at different concentrations, or with a more physiological stress, which is 40% oxygen instead of 20, which is a hyperoxy condition. So you can see you need weeks and months to see the same as you see acutely with 500 micromolar hydrogen peroxide. Let me get this one out of the way. That the TERT protein now not is in the nucleus, but outside. And if you co-stain with the mitochondrial dye, which is green, you can see now yellow dots, which means a TERT can go into mitochondria. And what it does there functionally, you can see in this slide. So again, we have in black the normal fibroblasts, which don't have any TERT or telomerase activity, either under normal conditions where it decreases oxidative stress and even more under higher stress. It also protects mitochondrial DNA, so you have more damage in the normal MRC fibroblasts and you have less in the TERT containing cells, either under H2O2 or again under hyperoxia. And importantly, it also promotes the resistance against apoptosis, again, either after H2O2 concentrations, again, you see the H-TERT overexpressing cells in red, or etoposide, which is used as a chemotherapeutic agent, for example, in breast cancer. So that's been published a long time ago. And we were now interested, because this was a cell system, whether it can also have some influence in the brain. So we did some experiments where we showed either mouse neurons, if you cultivate them from embryos in the lab. So after 2 weeks, they don't express any telomerase activity anymore. And also mouse brains can have telomerase activity into postnatal stage of around 3 weeks. However, during brain development, and this is an important difference between mouse and human, you already have a very early downregulation of telomerase activity, essentially after post-conception week 8, because there is nothing measurable, and 0.2 is the cutoff line. So everything below is nothing, and you can see also nothing in the adult human brain. So, however, in contrast, the TERT protein you can still see in old brains, and we used here brains from hippocampi of controls, which we used for our Alzheimer disease study. So they were in their 80s or late 70s. You see either histochemistry in brown here, the neurons which stain for TERT protein, or again immunofluorescence here in green, co-stained with neurotrace to make sure they're really neurons. You also have something in microglia cells, which are derived from macrophages, and it's known that they are positive for telomerase, but you don't have anything in astrocytes. And similarly, in cultured mouse neurons, you have TERT protein again in neurons, in microglia, but nothing in astrocytes, because you can see there is no co-staining, and this is just to prove the specificity of our antibody. This again was published 7 years ago. So we were interested to find out whether there's any protective effect of TERT against neurodegeneration. And I already mentioned our study of Alzheimer's disease, which I start with. And then we progress to our recent study on a mouse model of Parkinson's disease. So this is our Alzheimer's study, where we initially, our hypothesis was, because we knew in mouse brain it declines with age, at least the TERT gene expression. However, we didn't find any differences between Alzheimer's brain and age-matched controls, which means there's no influence of the disease on the TERT levels. However, if we now measure the mitochondrial localization in different hippocampal regions, we found that at least in CA1, 2, and 3, in Brach stage 6, so full-blown Alzheimer's, you have more TERT within the mitochondria. And you remember my second slide, where we showed that the shuttling of the TERT protein can be induced under oxidative stress. However, what we don't know is whether it's rather a consequence of the disease, or whether it's involved causally in the disease progression, because that's very difficult to see at this stage of postmortem analysis of brains. So what we also found very surprisingly was, if you look for control brains, you see they only have TERT protein shown here in red. However, if we take higher Brach stages of Alzheimer's disease, where we measure activated or hyperphosphorylated tau protein, which can form neurofibrillar tangles, we don't see any co-localization between those tangles and TERT protein, which can mean either that those neurons which have high levels of TERT don't succumb to any tau hyperphosphorylation, or hyperphosphorylated protein displaces TERT in proteins. And since it's easier to analyze that in a animal or culture model, we used mice, wild-type mice shown here in blue, which have normal expression of TERT and also TERT protein, and TERT knockout mice, which are shown here in red. And we transfused them with the mutated tau form P301L. And if we analyze now oxidative stress, which is shown on this graph, we see that at least in the dendrites, we have much more ROS reactive oxygen species in the TERT knockout mouse, in the neurons derived from the knockout mouse, which means the TERT protein seems to protect against oxidative stress, which is consistent with other results we and other groups found. And also the lipid peroxidation level now we see more in the neurons from the TERT knockout mice in the soma, which again is evidence for that in normal cells where we have TERT in the brain and in the neurons, that can be protected. So we took it from there and thought, okay, once we know that TERT might be protective in the brain against neurodegenerative diseases, we used two telomerase activators, which are essentially derived from a long-known Chinese medicine. It's from Mongolian milkweed, or the Latin name is Astragalus memornaceus. And we have two of these activators. So this TA65 is a very highly enriched plant extract. And we also used the chemical derivative from the cycloestrogenol. So in this plant, there are different components. So I'm not a pharmacologist, but yeah. So we wanted to use both, and they are both licensed to the company Neuroscience in the States in New York. So what we started with is and I apologize if you can't see the heading, but I don't want to do anything in case something disrupts. So we treated 2-year-old mice for 3 or 4 months with the 2 telomerase activators. And first, I show again what I mentioned before, that the TERT expression in mouse brain decreases with age. That's something we really haven't analyzed in human brains, because there are not many young brains available. And if we treat these mice now for 3 to 4 months on a daily dose with oral treatment, so we give it together with powder chow, you can see both activators increase the TERT expression level substantially. And if we now have a functional readout, so we put these mice, which are 2-year-old is quite old, so comparable to our 80-year-old PD patients, on a static rod. So young mice, of course, can stay on this rod without any problem. Why old mice? So these are the DMSO controls, because the activators are dissolved in DMSO. You see they can barely stay half of the time. However, if we now give at least the synthetic telomerase activator, they are almost indistinguishable from the young ones. And the TA65 probably has also an effect, but it didn't reach significance. So in order to also make sure that really the neurons are the cells which respond to the 2 telomerase activators, we cultured again neurons from embryonic mice, from wild-type mice, treated them with either the DMSO or the GRN and the TA65. And you see both after 2 days of treatment, probably we could have done only 1 day, we see an increase in the TERT expression. So I don't have to introduce again Parkinson's disease, because Huang already did this. Just to remind you, because it's important for my ongoing talk, that we have alpha-synuclein aggregates, which are supposed to play a major role, in addition to the decrease of dopaminergic neurons and function. And one of the important, or one of the many symptoms, but one of the very characteristic, is the tremor of Parkinson's patients, but you can also have, in some cases, the dementia. What is also known is that both mitochondria and autophagy dysfunctions can be involved in the disease. So we used a transgenic mouse model, which was generated by Elysée Maslaya, who is working in the United States at the NIH. And you can see this mouse model, it expresses human wild-type alpha-synuclein under the control of the PDGF promoter. And you see it forms the same alpha-synuclein aggregates, or also called Lewy bodies, as we see in human Parkinson's disease brains. And the highest expression you find in the hippocampus, the neocortex, and the olfactory region. So we focused our analysis also to hippocampus and neocortex. And what Amschel and colleagues found very important, that the increase of the alpha-synuclein, which is in the transgenic mice from the very start, only seems to increase at the age of 1 year, so 12 months, which seems quite comparable to the fact that most idiopathic Parkinson's disease also occurs in older patients. So, and this is in the hippocampus, but also in the striatum. And of course, dopamine levels also decrease in this model, so it's a quite valid model, and tyrosine hydroxylase activity as well. So if we treat these mice now, we treated them for 14 months. We started at 4 months because we were not exactly sure when the symptoms appear. We didn't want to miss this window. So we finished this study when the mice were 14 months. We analyzed both genders separately and jointly, so males and females, and we found in females, both activators significantly increased TERT expression. In the males, we only found a significance in GRN 510, but a strong trend in TA65, and we had only 3 mice here, so that might be a number fault. So if we combine both sexes, you see it's significant in both with both activators. So that's good news, and this was published last year in Progress of Neurology. We performed various behavioral tests, which we think are a good readout for what happens in Parkinson patients. However, when we use this rotor rod, where you measure speed, the time the mouse can stay on the rod, and the distance traveled, we found a very intriguing sex difference. So namely, in females, only the TA65 improved all parameters, while in the males, it was exclusively the GRN 510, so the synthetic cycloestrogenol. So we don't know exactly why, and we didn't analyze that because the study had to finish. But we assume that there are subtle differences between male and female brains, not only in humans, but also in mice. So, for example, it has been shown that there is different gene expression, etc., etc. In other tests, like when we measured walking speed, which is a readout for bradykinesia in Parkinson's patients, we again found a significant speed up, so decrease in the time the mouse needed to travel 1 meter, and with the TA65. However, this time in males, we saw an effect with both. So we then did a gait analysis or a stride length test. So we, while you do this with a computer and Parkinson's patients walk over a mat which is computer controlled with electrodes, we did a very low maintenance approach. So we painted the hind feet of the mice with 2 colors. So the right foot is painted with blue and the left foot painted with red. And as you can see, the control mice, they are very wobbly. They do long strides, they do short strides, while when you treat them with the 2 components, surprisingly, you see a very even stride, and also the length of the stride increased, and you can see the quantification below. So we did, we analyzed both feet, but only show you the left one because they are essentially identical. And again, oh no, I show both left and right. Both compounds improved the stride length. And as I already mentioned, the variation of the stride, which is very important, decreased, so that the strides are much more even, again, in the left and the right. And these are females, oops, sorry. And in the males, it's pretty much similar. And again, stride length and variation of the stride length changes. So stride length increases and variation decreases. So we also tried some readout for cognitive parameters. However, we couldn't do a bounce maze, which we normally would use for this type because of the locomotion differences between treated and untreated. So we used instead a novel object recognition, where the mouse sees the identical objects first, and then one is exchanged for a new object, and you expect the mouse to be more interested in the new object. And indeed, we see this for the females with both activators. However, for the males, again, only with the GRF AN510, which is very similar to that what we saw with the rotor rod. So as I showed you in the very beginning with our cell culture model, or also with the AD study, our initial hypothesis was that TERT, which resides in the mitochondria, might improve certain parameters. So we isolated mitochondria from brains and used amplex red test, where we measure the hydrogen peroxide, which is released from the mitochondria. We combined both sexes here, and we only saw, for some reason, an effect of TA65, both on the forward flow and the reverse flow, where we use a different substrate, the succinate complex for complex 2, versus pyruvate and malate for complex 1. So since we saw different behavioral tests, like, for example, the important stride length test, where we saw an effect of both activators, we don't think that the mitochondria are the important mechanism here. Unfortunately, there is no antibody which works in mouse brains, so anti-TERT antibodies, so we couldn't directly test how much TERT goes into the brain mitochondria. However, what we did is we analyzed the alpha-synuclein forms. So in green, with a secondary green antibody, you see total alpha-synuclein. In red, we have phosphorylated alpha-synuclein, because you might remember that in Parkinson's disease, there are different stages or different forms of alpha-synuclein. And these are the control mice. You see here the co-localization of green and red, so you see a lot of yellow. These images are all taken at the same exposure, and as you can appreciate, GRN510 decreases both alpha-synuclein types. In TA65, you hardly have any phosphorylated alpha-synuclein, but you still have a little bit of total. But in general, I think you will agree with me that there is a dramatic effect on decreasing both alpha-synuclein levels in the brain. And if we quantify it now, sorry, the slide is a little bit busy, and we analyzed hippocampal region CA1 and CA3, as well as neocortex, for the total alpha-synuclein level, which decreases in all treatments, while the phosphorylated is only significantly decreased after both activators and CA1, while in CA3 we have an effect only with TA65, because we have some outlier here. In the neocortex, we also see a decrease with TA65. So in this case, the natural plant extract seems to work better. And then, so this is phosphorylated is related to the DAPI. But if you now relate the phosphorylated to the total, because if you already have a decrease in the total alpha-synuclein, of course you will have less phosphorylated alpha-synuclein. But even if we normalize that to the already decreased level of total alpha-synuclein, we still see significant decrease with the TA65, the plant extract, both in the CA1 and in the neocortex, but we don't see it in the CA3. So in order to be sure that this has nothing to do perhaps with fluorescence, with our applied fluorescent method, we also used the well-known immunohistochemistry, where we use another antibody and we use a substrate, which is shown here in brown. Now we can only analyze the molecular layer and not the processes of the neurons here. But so you will see the brown dots here, which you can appreciate are getting less with the treatment with GRN510, and there's hardly anything with the TA65. And you see this significant decrease here quantified with immunohistochemistry, and we analyzed the same molecular layer in our immunofluorescence staining, and you can see both methods work confirm each other very well. So this means it's really true. It's not anything to do with a fluorescence artifact, and we were very happy about this. And last but not least, of course, it's important to analyze also aggregated alpha-synuclein, which we co-stained again with the total alpha-synuclein. It's shown here in the neocortex. And when you overlay or merge both stainings, you see a lot of yellow. However, again, with the treatment with GRN or TA65, you hardly see any total. So you see a little bit, but you hardly see any aggregated. And if you quantify it, for some reason, it's only in the CA1 significant with TA65. But this time in the CA3 region of the hippocampus, it's highly significant with both activators, and the same strong decrease, and you see it maybe only 50% occurs in the neocortex. So of course we were now interested why this might happen, and what we know is that protein degradation mechanisms like proteasomal degradation or autophagy mechanisms are decreased in the brain with age. Maybe not only in the brain, but it's been well researched in aging brain and also during neurodegeneration. And so we analyzed P62, which is an important adapter protein of autophagy, but also the LC3B, which is located on the membrane of the autophagosomes. And we found that at least in the CA1, both components strongly decrease with treatment. However, in CA3, only the P62 decreases with TA65. In neocortex, we have some trends, but no significance. So these are essentially pilot data, but we think that it's not surprising that important protein degradation methods like autophagy are maybe involved, and other people have shown that TERT might even form complexes with, for example, mTOR, which is upstream of autophagy, at least macroautophagy. So we think this stimulation of autophagy might occur due to the telomerase activators. So to summarize my talk, I try to convince you that treatment with telomerase activators, which in our case, where neurons don't have telomerase activity, they are just increasing the TERT expression level. But as I told you in the very beginning, the TERT protein has a lot of important non-canonical functions which don't have anything to do with their enzymatic activity, which is related to telomeres. And neurons are postmitotic cells, so they don't shorten their telomeres because they don't divide. So we showed in the study that the treatment with the two telomerase activators, GRN 510 and TA65, could improve movement, balance, and gait in our alpha-synuclein transgenic mouse model. And analyzing brain pathology, we found that all three forms of alpha-synuclein, total, phosphor, and aggregated alpha-synuclein, decrease substantially. And we think that the mechanism behind this decrease is that the TERT, which often is used as synonymous for telomerase, but without activity, that it activates protein degradation mechanisms such as autophagy. So, and in my acknowledgement, I want specifically acknowledge my then PhD student Teng Fai Wan, who has now finished his PhD and is working as a postdoc. I had MSc student from the UK, Emma Veer, and previous data were generated from other people in the lab, Shahida Ahmed, Alison Spielsbury, and Abbas Ishak. And I also thank TS Science for providing the two activators, and my collaborator for autophagy, Viktor Kowalczuk. as well as the pathologists Johannes Adams and Mary Johnson. And I thank you for the attention, and I'm happy to take questions.